is Dr. Mary Tavares.
I'd like to thank the committee for the opportunity to offer the perspective of the dental research community as you examine the 2009 FDA ruling regarding the safety of encapsulated dental amalgam. My name is Dr. Mary Tavares. I am a senior clinical investigator at the Forsyth Institute in Cambridge, Massachusetts, and a board-certified public health dentist. Forsyth is a nonprofit independent research organization dedicating to advancing oral health through scientific discovery and education. We are affiliated with Harvard School of Dental Medicine and Harvard Medical School. Today, I'm here representing the American Association for Dental Research, a nonprofit organization whose mission is to advance dental and craniofacial research. The 2009 FDA ruling concluded that, and I quote, "Clinical studies have not established a causal link between dental amalgam and adverse health effects in adults and children ages 6 and older. In addition, 2 NIH-funded clinical trials in children aged 6 and older did not find neurological or renal injury associated with amalgam use." As co-principal investigator of a US-based clinical trial referenced by the FDA in their final rule, I support the agency's decision to classify dental amalgam as a Class II device. The New England Children's Amalgam Trial that my colleagues and I conducted was a randomized clinical trial to evaluate the health effects of dental amalgam in children. During our study, 500 children between the ages of 6 and 10 who had never had amalgam fillings but were in need of treatment were enrolled into clinics from sites in Boston, Cambridge, and Farmington, Maine. They were randomly assigned to receive either amalgam or composite fillings, and we followed them every 6 months for 5 years, collecting blood, urine, hair samples, and neuropsychological testing. The final results of this trial, as you've heard before, were published in the Journal of the American Medical Association in April 2006. The conclusions were that no statistically significant difference in adverse neuropsychological or renal effects were observed in the children whose teeth were restored with dental amalgam as compared to composite resin. In the final report, our team concluded that under the conditions of use represented in the trial, there was no reason to discontinue the use of mercury amalgam as a standard of care for caries in posterior teeth. This is particularly important consideration for areas both in the United States and in other countries where the replacement of mercury amalgam with composite material may not be feasible with respect to factors such as cost, storage, and expertise in handling, and thus could adversely affect the dental as well as general health of the public. The findings from the 2006 study indicated that the health effects of dental amalgam need not be the basis of treatment decisions when choosing a restorative material. This is a key consideration for those of us in the field of public health. Absent any clinical evidence that amalgam would cause harm and scientific evidence confirming the safety of amalgam, a speculative limitation on its use would certainly impact access to restorative dental care for many populations. Mechanically, dental amalgam is in several ways a better material than composite with an overall lower cost of treatment.
You will need to wrap up in the next 20 seconds.
However, composite is less durable and needs to be replaced more often. That was also a finding from our study. As I conclude, I want to re-emphasize that there are no scientific peer-reviewed studies found a link between amalgam restorations and any medical disorder. The AADR commends you for your thorough review of the peer-reviewed scientific literature and for arriving at a decision to permit its continued use. Thank you.
Thank you. Thank you for that presentation.
Unfortunately, I've forgotten the name. Is the speaker from the New England study still here? Thank you. I was just wondering, do you have data on the subjects, the maternal amalgam load?
I'm Dr. Mary Tavares. We don't have data on maternal. We have yes, no, I think, but I'm not 100% sure. But we did not examine the mothers, no.
So there's no data on amalgam exposure as well as the fish consumption?
We do have fish consumption data. I don't have it with me. We have dietary data, extensive dietary data. We have the methyl mercury from the hair analysis, but we don't have, and we have extensive medical history, but my recollection is we don't have detailed maternal amalgam exposure.
Thank you very much. Well,
Thank you, Dr. Ginsberg, for raising the Echeverria studies. I noticed that the tests, the neuropsychological tests they used, also used in the two randomized. There was a lot of overlap with the two randomized children's trials, the Echeverria and the New England study. But those children's trials were negative in those
Right.
for those tests. Can you rationalize that
Right.
apparent discrepancy?
Certainly, yeah. That is a good point, because if we're worried about these low dose effects, then why were those studies negative? Well, there's the issue of the length of the window of maximal sensitivity in children being more the perinatal period, likely, rather than when the CNS is more developed, which these studies started at age 7 or 8. The follow-up period is 5 to 7 years in those studies, so the full effect of amalgam exposure on neurological systems would not be expected to be manifest at that point, although it is a very useful design to do that. Probably more follow-up would be a good thing. But bigger than that, and a bigger issue than that, is that those studies, I don't feel, adequately corrected for the amount of background exposure to methyl mercury that could be coming in the diet. Certainly the full amount of, for example, the Cassiopeia study had urinary mercury data, and they did not test the correlation between urinary mercury data and their endpoints. And I've been talking to Diane Echeverria at the University of Washington. They were part of the Cassiopeia study. They have the raw data, and they are looking at those correlations now, not just for the porphyrin effects, but also for the neurological endpoints, and they're writing those results up. So I really encourage FDA to look at reanalysis of the Cassiopeia data based upon urinary mercury exposure, which is a composite of background diet as well as the amalgam exposures, to see whether those correlate, to see in a peer-reviewed journal publication whether those correlations hold up from what I have a personal communication about. Same thing though with the Boston study. That they had populations that had similar amounts of dietary methyl mercury exposure, the amalgam and the no amalgam group, but they didn't do that wasn't part of their continuous logistic regression model. So it wasn't properly controlled for, in my mind, the amount of background mercury exposure in the Boston study as well. And a major confounder would be, as I talked about, background exposure to methyl mercury, this being layered on top of that as an additional risk.
Just
To discuss whether the FDA appropriately represented the strengths and weaknesses of the available clinical data, I think we can say no. We don't think they have. And we can maybe give them some areas where maybe the strengths are overstated or understated, and where the weaknesses are overstated or understated, but I don't think it's worth our while to try to wordsmith everything for them.
I find it interesting, in a label, you are actually referring to two clinical trials which have already been brought up this afternoon who had weaknesses, and yet the way the sentence is worded, it's very definitive. And I just wonder if, in terms of, I would ask the FDA to perhaps consider reconsidering how that's written, because they say did not find any neurological, and there's already been some assessments of the measurements used had some limitations, and renal injury, which perhaps has some limitations. So I'd ask that you modify that sentence if you want to leave such a sentence in a label, which is a little unusual to refer directly without referencing the study.